Hi Year 11, this is Mrs. Chips, hope you're all doing alright. Just going to try and create a little video here to explain some of the key bits from genetic modification, which is quite hard to do just from the booklet. So we'll start with key bits to do how we actually do the genetic modification, and then we'll look at how we can identify which bacteria have been modified. Now it's worth noting when we're talking about genetic engineering and genetic modification, same thing. And also that most of the time we're talking about using bacteria for this because they're much easier to manipulate, and we can easily add additional DNA to bacteria through the use of plasmids. So we'll just quickly recap bacterial structure. So you are supposed to know already about the structure of bacterial cells. So hopefully this is familiar to you. But we have here bacterial cell. It's got a cell wall and a cell membrane. It could have an extra layer on the outside here called a slime capsule, which is just a protective thing. It might have some flagella. We can add them on there. It doesn't matter too much, but the key thing here is that the bacterial DNA comes in two forms. So you have this kind of almost like a balled up bit of DNA here, which is known as the chromosomal DNA. So this is the main part of bacterial DNA. But bacteria also contain small rings of DNA called plasmids. And these are the bits we're interested in when we're looking at genetic engineering. So plasmids quite often will contain genes to do with antibiotic resistance. So these are bits of DNA that are often passed between individual bacteria, so they're almost like sharing their antibiotic resistance genes. And because of this, it means that we can actually take these plasmids, we can edit them, and we can insert them into bacteria, and the bacteria will produce whatever proteins that bit of DNA codes for. So we can manipulate this to make the bacterium produce what we want. So say we've got a human gene that we're particularly interested in. Let's just represent it like this. Let's say this is the insulin gene on our human DNA. If I can get this gene into a bacterial plasmid, then I could insert the plasmid back into the bacterium, and I could create a population of bacteria that all produce the insulin that this codes for. So let's colour this in. So we've got an insulin gene there, and I've got my plasmid. So this is just a ring of DNA from the bacteria. And what I can do is I can cut a certain point on this plasmid, and I can then insert this insulin gene in there. So I can end up with a plasmid like this that's mostly made of bacterial DNA, but it has added into it this extra bit of DNA from the human. So we have a plasmid containing a human gene. So I can then put this back into my bacteria, and then as the bacteria multiply, they produce a population that all contain this plasmid with this human insulin gene. And so one of the products these bacteria will make is human insulin, and we can then collect that off, purify it, and we can use it in treatment of patients with diabetes. So that's the general gist of how the process actually works, what we're aiming to do. But within your booklet that was set last week, there's a question that takes this a step further, and is thinking about how we can identify which plasmids have successfully been modified to include the extra gene. Because as much as we can plan out this process, it doesn't work perfectly. And actually when you do this in a lab, you get lots of plasmids that maybe get cut and then they re-stick together, or they get cut and added to another plasmid themselves, and then you'll get some that are successful where they've been cut and the new gene has been added. Those of you who go on to do A-level biology, you'll probably get the opportunity to actually do some lab work where you do this process. Certainly those of you who stay with us will do, and so we'll go into this in more depth at A-level, and you'll get hands-on and do this work. So we'll have a think about how we identify these plasmids that we want. So the bacteria that we produce can contain these. And this is all to do with these antibiotic resistance genes that the plasmids contain already. Because I can use this to identify which bacteria contain plasmids with these genes. So let's have a think about the example that was in your booklet. So in that example there was a plasmid like this which had 2 antibiotic genes on it. Antibiotic resistance genes, sorry I should say. So there was a gene over here and a gene over here. Obviously the positioning doesn't actually really matter. But this one was going to allow us to be resistant to tetracycline, and then can't even spell. The other one made the bacteria resistant to ampicillin. So there's lots of different types of antibiotics, and bacteria frequently carry these resistance genes, and like I said, they can pass them to each other, so they can end up with multiple different antibiotic resistance genes. The one you'll be most familiar with is MRSA, which carries amoxicillin resistance. Oh, sorry, not amoxicillin, methicillin. Anyway, so these give them a resistance to antibiotics. What that means is bacteria containing these genes would be able to live in a solution that contained these 2 antibiotics. Now with the insulin gene, the enzyme that we use to actually cut the plasmid is designed to cut in the middle of this tetracycline resistance gene. So I get a plasmid where that tetracycline resistance gene has been split. Now what that means is we've no longer got that piece of code in the right order, because there's going to be something else put in here, and that's our insulin gene. So the bacteria can no longer use this bit and this bit here to make the product that makes them resistant to tetracycline, which means it's lost its resistance to tetracycline. So no longer resistant to, I'm just going to call it T in this case. Let's put brackets T there, and we'll call this one A. But it does still have the ampicillin resistance gene, and that's not been interrupted, so it's still making the product needed to be resistant to ampicillin. So any bacteria that contain this plasmid in the form we want, so this is the plasmid we want, any bacteria that contains this plasmid will be resistant to ampicillin, but it's not going to be resistant to tetracycline anymore. So it's only resistant to A, not T. So just abbreviate it. Now there are going to be other plasmids formed in this process as well, because some of them will have come out like we want, but some, like we discussed over here, will have been cut here and then just refused back together. So there'll be some that still contain both resistance genes unchanged. There'll be some where it's cut here and it's fused to another plasmid, so we get like a extra big plasmid. It will still contain a version of this tetracycline gene that is unaltered, and it will still contain this. So, and we'll also get some plasmids formed just by the insulin gene sticking to itself. So you get a little loop of 2 insulin genes, or maybe 3 or 4. That obviously won't have any resistance to any antibiotic that we're using. So when these plasmids get taken back up by the bacteria, that's part of the process we do, those bacteria are going to be resistant to whichever resistant to whichever antibiotics they've got the genes for. So all the ones we want are only resistant to A but not T. That means if I create a little bacterial colony, so say I get my petri dish and I grow these bacteria in here, there's going to be colonies in various places that form here. And what we can do, that wasn't meant to come out of a smiley face, never mind. What we can do is we can get a piece of filter paper or a special type of paper, we pop it on the top there, and we lift off a sample from each colony, and we can then test those with antibiotics, or we could just test it directly in here. We can grow a plate where we've got our colonies growing in agar that has these antibiotics in. And what we might find is that some of these colonies survive. So we might get, let's just shuffle this down, we might keep a colony here, and maybe here, but we lose the other ones. And that would tell me that the colonies that were growing here and here and here had bacteria that were not resistant to both the antibiotics. And so I could use this to identify which colonies were only resistant to ampicillin and not to tetracycline. And then what we can do is we can grow bacterial populations from the colonies that showed the right genes present in terms of their antibiotic resistance. So then we know those are the ones that definitely have the insulin gene in. So then we can grow that colony into a big population. We can do it in an industrial sort of vat, and we can then collect off the product, which will be the insulin, and we can purify that and use it. Now, we could apply this to things other than just insulin, but this is the classic example. But essentially, if we've got a gene that allows us to make a protein for a product we want, we can do this process for it. So that's, in a nutshell, what I wanted to explain to you. Email me or contact me on Teams if you've got any questions. Hopefully, though, that covers everything.